Okay, now we're ready to start talking about enzymes, the way that we can make a reaction take place faster and with less input of energy than without the enzyme. So one thing to remember is that the delta G is always unaffected. It's going to always be the same amount whether or not you add the enzyme or not. It's just that the enzyme can lower the activation energy. So what is an enzyme? So here's a space-filling molecule of an enzyme. Enzymes are proteins. And remember when we talked about proteins, they have very specific 3D shape. This is the tertiary structure. This enzyme does not have quaternary structure because there's just one subunit. And it's usually folds into a shape to make a nice little pocket for the reaction to take place. Because the enzymes are very specifically shaped to only fit certain substrates. Sometimes if a substrate competitor maybe is a slightly similar shape, it could sneak in there and block this. But essentially there's only a certain substrate that can ever fit in the active site. So nomenclature that you need to pick up as we talk about this vocab. Enzyme's a protein that speeds up a reaction, catalyzes a reaction. The thing it's working on is called the substrate. Sometimes they talk about ligands, but substrate is the thing it's actually ripping apart. In this case, we'll talk about in a minute. The active site is where that substrate binds. So you can see here, sometimes when the substrate binds, the enzyme actually changes shape to help catalyze that reaction. So here's an example of two substrates that need to be put together, or maybe changed in some way so that in this case it looks like two other things escape. So enzymes can work to put things together. Enzymes can work to take things apart, and in this case that they're showing you here, and this enzyme is working to flip-flop something, maybe just end up making a different product. So again, substrates fit in, somehow they become unstable, a new bond gets created, and now we release something totally different. Enzyme is there to be reused. So enzymes are never used up in a reaction. So when we talk about our experiments that are going to come up in lab, your enzyme concentration always stays the same. You can hit a point where they're all filled up and they're working at capacity, but your enzymes never go away. So enzymes, because they're proteins, they have some pretty specific parameters that they function in. They are constrained by temperature. So if you heat up an enzyme that maybe belongs in a typical human cell, our temperature in Celsius is 37 degrees, it's 98.6 degrees Fahrenheit. If you put one of our enzymes that's maybe working in a skin cell into boiling water, 100 degrees C, it will denature and it will unwind and it will not work anymore. On the flip side, if you isolate an enzyme from a thermophilic bacteria, a bacteria that loves heat, it actually will survive at those high temperatures. And we'll talk about an enzyme that was characterized that we use in a reaction called PCR, where it was purified from a bacteria that lives in the deep hot vents. Sorry, my dog. Hot vents, and it actually thrives at high temperatures. We also have optimal pHs for enzymes. Again, they're proteins, so maybe a low pH would degrade a typical enzyme that works in your intestines, whereas an enzyme like pepsin that's designed to rip apart proteins in your stomach has to survive at a very low pH, and it actually has an optimal pH of like 2.8, it looks like here. Basically what these diagrams are showing you is the rate of reaction of an enzyme. So it's the rate of that enzyme is very efficient up here and very inefficient or lacking down here. We'll do a lab that's going to show you some of this stuff too. So you can, in addition to maybe boiling an enzyme to make it stop working, which is not a very good way to regulate something in your body, there are other ways that enzymes can be regulated in your body. There's something called a competitive inhibitor. So in this case, normally the enzyme can fit the substrate nicely in that little active site. In the case of a competitive inhibitor, as I was mentioning before, maybe that inhibitor has an affinity. Remember, these are all R groups sticking out here, so they have some chemistry going on. And that competitor can interact with those R groups and bind, and then block your substrate from going in there. It's called competitive because there is some concentration of your substrate that you finally can beat out this guy. I don't know if in chemistry you got to it. If you take AP Chem, you'll definitely talk about there's things called affinities, how likely something is to want to bind to each other. If you have a very high affinity, its on and off rate is very slow because it stays bound a lot. So if a competitive inhibitor has a very high affinity, it will not allow that substrate to get in there. However, if the competitive inhibitor has a very low affinity, just kind of bounces off, you can imagine that the substrate could sneak in when this guy is not bound. Then there's guys called chemicals called non-competitive inhibitors. Basically, they bind another site on the enzyme. See, there's this other location down here. And when that guy binds, when that chemical interaction takes place, you can shift the structure of the protein. Maybe an alpha helix shifts over. And now the substrate binding area is not effective, and it won't work anymore. These are usually non-reversible. So you can kind of take that idea and use it to regulate an enzyme in your body. Let's see if this zooms in a little bit more. Actually, I'm not going to do that. I'm going to go back here. So sometimes this is an enzyme that has 4 subunits, and it can be stabilized if this little green activator molecule interacts with the R groups that are hanging out in between here on that protein and stabilize it in the open conformation. Similarly, there could be an inhibitor that would stabilize this enzyme in a non-active form, an inactive form where that active site is closed up. And maybe this enzyme complex can oscillate differently in 2 ways between the active and the inactive, and you could freeze it in one stage or another. Similarly, there's another kind of allosteric activation where the enzyme exists in an inactive form, but the substrate comes in and then opens up all the other active sites as well. So now you have a more efficient enzyme working. All this takes place because proteins can change shape depending on what chemicals bind them. Very slight changes. Usually it's not a big massive change, it's a little shift. So this image shows you how you might want to regulate an enzyme in a living system. What they're showing you here is a 5 enzyme step process to go from threonine deaminase to produce isoleucine. Basically, you take threonine, it binds threonine deaminase. There's 5 different steps, and you end up with isoleucine. So you do not want to be doing this if you have tons of isoleucine around. So in this case, the isoleucine can go back and inactivate that original enzyme. So basically, you don't make isoleucine if you don't need it hanging around. If you have no isoleucine around because it's been used up by the cell, you need to make it. So now that it falls off because it's been used up, the active site's available, can bind threonine, and now we can do that. It takes a lot of energy to work through all those different intermediates and make those reactions take place. So you don't want to do it if you have a lot of that around. We'll talk about this really soon when we talk about glycolysis, the way we generate energy by ripping apart a glucose molecule. And if there is a lot of ATP hanging around, we don't want that reaction to take place, so we actually will stop the whole reaction very early. There's lots of places in our cells where there's incredibly high amounts of enzymes that are doing lots of different work, and the mitochondria is one of those places. So here's an animal cell. Mitochondria are the places where we make ATP. The electron transport chain is embedded in those inner membranes, and the Krebs cycle or the citric acid cycle takes place in the matrix. And basically the inner of the mitochondria is so full of enzymes it's almost like a jelly. We're going to talk about that in the next chapter. I kind of did that one already. Whoops. So let's talk about this example of the enzyme that rips apart sucrose. So remember, sucrose is a disaccharide. It's made of glucose and fructose. And although this is a spontaneous reaction, meaning the delta G is negative, you put sugar out on your table, it doesn't spontaneously in your lifetime fall apart, right? It's spontaneous in the exergonic sense, in that it will release energy when the bond gets broken. So in order for it to happen more efficiently in your body, we have an enzyme called sucrase that binds this disaccharide and splits it up using a water. Remember, that's a hydrolysis reaction. And separates it into the 2 monomers. And the next image should be a little YouTube video. Whoa, we don't want that to be noisy. So it's going to show us sucrase. It's happening, it works inside the intestines. And there's a picture of a model of the enzyme sucrase. And you see how it has some nice binding site here to fit the dimer and to fit the water molecule. So here comes the dimer. and it says that it changes shape a little bit when it binds. Water molecule comes in, and now we separate it into the two monomers. I'm just going to show it again. The active sites bind the glucose and the fructose. When that happens, a little tweak, that oxygen bridge gets opened up to react with the water molecule, and then that's where the hydrolysis takes place. So, you can go back and watch that whole thing if you want, and I think that's the end of the show.